Hi, today we're going to be talking about protein synthesis. This is also called translation, and this is the point in the outline where we are. So one thing to start with is that the main thing that we're trying to do here is we're trying to take the DNA and the code that is on DNA and translate that code into a protein. We've talked about this before, that the proteins are mainly enzymes in the cell. They are also structural, of course, and so forth, that we talked about earlier on. But the proteins are what really gives a cell, and indeed an organism, a lot of its characteristics. In other words, the traits and things like that we've been talking about in terms of genetics. And we have to translate that DNA code into the protein. But one of the problems is we have 4 nucleotides, that is adenine, thymine, guanine, and cytosine, and we're going to translate that into 20 amino acids. So this is kind of like taking numbers and representing the letters of the alphabet. For example, 1 equals A, or you could say 0 even if you wanted to, 2 equals B, so forth down the line. And we have 26 letters, and that they need to be represented then by more than one number, right? In other words, once we get to 9, we have to go to 10, and that would be 2 numbers representing 1 letter, right? So this would be the same thing as once we get past 4, then when we get to 5 nucleotides representing an amino acid, we have to go to 2 of them, right? And it turns out that that the number that we actually need to make 20 amino acids is 3 nucleotides in a sequence is going to equal 1 amino acid. Now, if you figure this out, if you want to do the math on this, it's like we have 4 nucleotides, and 4 to the 1 gives us 4 possibilities, so that's only 4 amino acids that we're going to come up with. If we had 4 nucleotides, and we use 2 of them at a time, for example, A and T, or A and G, or A and C, something like that as a code, then that would only give me 4 to the 2, or 16 possibilities. So instead, I do 4 to the 3, that is 3 nucleotides in a row, and those are going to give me 1 amino acid, and that makes plenty to go around, because that would be equal to 64. All right, so we have 64 possible codes. That means there's some redundancy in the code, because I only need 20, actually, but there's going to be some that are representing other things like where to stop, where to start, etc. And again, some codes, there's going to be more than 1 code for 1 amino acid in some cases. So this is the way it works, all right? So this is looking at a piece of DNA. You might remember that, of course, we have the nucleotides that are here in the middle. This is the sugar phosphate backbone on both sides. It's a double helix. Hydrogen bonds in the center. The sugar phosphate is covalently bonded on the side. And what is happening with a particular gene is a gene is going to be read on one side only. And it's going to be read in a particular direction. It will have sequences of nucleotides that'll say this is the beginning. It'll have sequences that say this is the end. But basically, let's say, for example, it's going to be read from here to here. That is down to the bottom here. So I'm reading in one direction, and when I do that, what I'm reading is I'm reading 3 in a row. So 1, 2, 3. So this code or codon would be AGG, and that's going to represent 1 amino acid. And then the next one in the sequence would be CTA, and that's going to represent the next amino acid in the sequence. And then there's going to be AGC, that would be the next amino acid in the sequence, and CTT, the next one. So in this particular gene, which is ridiculously short, I would have 4 amino acids in the sequence, and it would be 4 amino acids long. So that would be the coding information for 1 amino acid per the 20 possible amino acids in a protein. So just to remind you about proteins that are made of these 20 amino acids Remember, they have the primary structure, which is the sequence of amino acids in a row. And then they have the secondary structure, which is the folding, and the tertiary structure, which is the globbing. Most of the proteins in our body are globs, and some of them are quaternary structure, which is they have more than one tertiary stuck together, like hemoglobin, for example. All right, so just remember that those are what we call functional proteins. The tertiary mostly are what are functional proteins, and the amino acid sequence alone is just the linear structure, or what we call the native protein.